Yeah, I have to thank the organizers as well to invite me here. And I have the task to keep you awake for the next 2 hours or so. Yeah. You pay me, huh? Okay. At the end of the first day, for this purpose, I have made some questions. You see here some questions that are a little bit provocative, but probably at the end of my talk you will understand what I mean with this. If we rethink occupational limits or other regulations on the basis of weak studies. So this is, to my opinion, a very, very important point. And you see, this cartoon is 15 years old. I showed this nearly 15 years ago. The situation has not changed. It's all the same. So, and I believe that our students from today have enough to do for the next 50 years on this issue as well. Okay, some facts related to what Len has mentioned already. So he set the stage for occupational exposure limits very nicely. So we now go to some values, and based on these facts, so we have a lot of titanium dioxide that is produced, and only a small amount, 40,000 tons, if you declare it as small amount, is nano from this overall. What the people don't know either is that the nano titanium dioxide protects us from skin cancer, because it's a very nice UV blocker in sunscreens, much better than the chemicals that we need there. And some of these are endocrine disruptors that are distributed into our waters. And so the titanium dioxide is probably much better because the environmental background is much, much higher. So we have nano titanium dioxide in the water, in the sediment, and in other environmental compartments as well. And the next point is what I will tell you about a little bit during my talk, is that most of the studies, especially those that are on DNA damage, between the years 2000 and 2013, deliver weak results, flaws in the study design, and wrong conclusions. And most of the evidences that are reliable show into the GBP direction for titanium dioxide. This is already mentioned. You know this NIOSH paper, and based on this, you see here in the first row the NIOSH recommendations and the OSHA recommendations, and you see that, no, the NIOSH recommendations come on the next slide. You see here these exposure limits for the titanium dioxide that we had so far. You see that the MAC Commission has not given any value. Helmut has explained this because there is no categorization between the two. But for the particles not otherwise specified or regulated, we have these for the respirable particles, we have the 1.5 milligrams per cubic meter. And what the NIOSH did here in the first row is they discriminate between the fine, the normal pigmentary titanium dioxide, and the ultrafine titanium dioxide, and you see here the 2.4. This is mainly based on these 0.5 milligram that we have for the GBP, but reflecting a density of 1 for the material. So with the 2.5 density, we have 1.25 milligrams per cubic meter. This is the OEL, and titanium dioxide with the 4.2 or 4.5 density, so we have 2.4 milligrams per cubic meter for the pigmentary. And the NIOSH recommended for the nanoparticular titanium dioxide these 0.3 because of the higher surface area that we have. We have heard about this. And you see the MAC Commission, also they haven't given a value, recommend that it is not different to the normal general particle effect of GBP so far. There are some other papers. One is reflecting here Jan. It's a Polish paper in Polish language, but the abstract was in English. And you see here that they recommend the 0.3 milligram for the nano titanium dioxide as well. And there is a very recent European project named Scaffold, and they recommend the 0.1 for the respirable fraction. So this is only to give you some numbers, what is now in the air, what are we discussing about. And this is the wrong one. And this is a new slide I put in today because we had this, in this morning, we had this discussion. And this document is only in German. It's not translated so far in English language. So in English you have the older version from 2013. And this is for manufactured nanomaterials. This is the announcement for the hazardous substances. And on the next slide, or on the next page, I have here the paragraph 4. And you see here, it is still this exposure limit in this year, June 2016, of 0.5 milligram per cubic meter on the basis of 2.5 grams per cubic meter. You have then 1.25 as an exposure limit. So, and with this, we go further to the studies, to some of the studies, and I made an evaluation, a very deep evaluation of the literature of the last 15 years, back to 2000. You see here the increase on nanotoxicology during the last years. I closed my study here in May 2013, and up to now we have 25,000 papers on nanotoxicology of different materials, of course. When I did my study and collected the literature, I had 12,500 papers. At the end, I selected, of course, for PDF and for language and for other reasons, so I end up with 6,000, more or less 6,000 papers. You can find the first citation here, and you can have some more details on the literature there. And we have a lot, of course, coming along with the European funding program, with the national funding programs that are stated here. So we have a lot of money put into the overall scene. And I have here the next provocative statement. Just now to say that we have not enough information about the hazardous effect or other effects, biological effects of nanomaterials is wrong. And I hate if someone starts his paper with, in the introduction with, as we don't know enough about this, we have made this study. This is, I reject this normally. Okay, it's not true. But what is true is that most of the literature of these publications are not good. So they have shortcomings and other things. We have a German funded project. You find it on the web. It's a web portal for nanomaterials. It's the DANA project, nanoobjects.info. And on the knowledge base here, if you click there, next, of course, you find the supporters. This is the German Ministry for Educational Research and the Swiss government as well. They give some money for this project. And here we have a literature criteria checklist, and you can download it. And this literature criteria checklist is the basis for the literature that we select for our data that we present on the website. And this contains mandatory and desirable characteristics. These are the mandatory ones. And you see here some very important things. First is the physical chemical characterization, some very important points. Not all, some are desirable. We take some only, so not all. So this is the one that is very important. Then the next, the sample preparation is of course very important, and the dispersion. And here are the 4 points where most of the studies are rejected. Because it starts already with controls, missing controls. Most of the studies don't have any control. Many of the studies don't give the right concentration that has been used. I come to this point later. So you see here many reasons for flaws. And this is the study, the first part of this study that I have published. It's open access in the Angewandte Chemie, and you can download it. One of the results that I have found is this one here. It's in vivo, it's a skin, because nano titanium dioxide in cosmetics is one very important issue. Therefore, the transport through the skin is very important. And you see here that only a very few number of studies have found nanoparticles in the body, so-called, after applying on the skin. These 5 studies, if you look carefully into these 5 studies, these are not reliable. So most of the studies demonstrate that there is no transport through the skin. And now I tell you a story about this skin. You see here a paper. This is Chinese Whisper. Yeah, you know this game? Chinese Whisper. So you have this first message. You whisper in the ear of your neighbor. The neighbor has to whisper the same message the next. The more stages you have in between, the more funny is the end of this message. And you see, I have an example. You find here titanium dioxide. It's the Evonik P25. And it produces reactive oxygen species in microglia cells that are isolated directly from mouse brains. And these cells are in vitro exposed. No matter, of course, they produce reactive oxygen species because macrophages have to do. It's no toxic. It's a normal process, okay? 2 weeks or 3 weeks later, the Nature block comes up with nanoparticles in sun creams can stress brain cells. Referring exactly to this paper. Yeah, okay. So this was the first flaw, because they didn't mention that it is the sunscreen that is doing it. They only say that the particles, titanium dioxide, are doing this. And then the German paper, Taz, came up with the headline, "Sun cream can destroy your brain." If you don't understand, I have transferred it into English, so sun cream can disintegrate your brain. So never use sunscreen, please, with titanium dioxide, because then your brain is not functioning anymore. And a little bit later, the Mirror of UK comes up with this. This is what I mean with Chinese Whisper. You start a story with facts, and you end up with fairy tales. Okay? And this is true for most of the studies that you find in headlines in the web. This is true for all these. Okay, coming back to real science. Uptake studies, I mentioned already the skin version in yellow. Ingestion and inhalation, you see that there are studies that demonstrate an uptake. And this is clear because nano, and this is only nano, this is not pigmentary titanium dioxide, nanoparticles will be taken up by any cell. If you expose a cell to nanoparticles, you will find nanoparticles inside of the cell. It is not possible that the cell can avoid this, because they have vesicles where they take up material from outside, they have to feed themselves, they have to drink, and with these vesicles, of course, nanoparticles can come into the cell, so it doesn't matter. And this explanation you already have seen, the discrimination between primary and secondary genotoxicity. And I looked in all these studies for, this is a selection for titanium dioxide, for these different effects. And you see the studies on genotoxicity in this range, 2000-2013. You see that there is no single study that shows primary genotoxicity, only secondary. And even in these studies, in the overall number of studies, not all show and demonstrate genotoxicity. And if you look exactly for these 40, the yes and the no studies, they build up these 40 studies here. The 40 studies, they have investigated genotox effects. Only 22 showed a positive effect. From these 22 that showed a positive effect, only 12 have measured in a concentration range below acute toxicity. Because for me, it makes no sense to make genotoxicity assays in an acute toxic dose range or concentration range, because anyway, the cell will cleave the DNA during necrosis or apoptosis. So it makes no sense to make a COMET assay with such cells. But they do it. They make a COMET assay in apoptotic cells. So this is nonsense. Who is financing these things? So, okay, and from these 12 studies, only 4 studies have used a minimum 2 different tox assays to be sure that the range of toxicity is the correct one. Because we have shown that especially nanoparticles, because of their large surface, they interfere with the MTS assay, they interfere with other dye assays, neutral, red, and so on. So they interfere with your assay system and show you false positive or false negative effects. Therefore, you should have a minimum 2 or more tox assays. So that the rest is only 4 studies from all these studies that came into this. And I do not judge the, let's say, the reliability of the studies. This is only the starting point if they rely on the criteria. If the data they present are then reliable is another question. If you look for inhalation and instillation, what we are doing here for titanium dioxide, pigmentary or nano, you see here, these are the studies that have been done in that. The red ones are the instillation, the blue ones are the inhalation studies. And you see that there's no primary genotox study in animals so far. There's only some that are related or refer to quartz-like effects, so a severe inflammatory effect, a severe oxidative stress effect. Most of the studies show that it is like a GBP. And there are also 9 studies, instillation as well as inhalation, that show no effect. And you find, of course, a lot of studies that have overload conditions, especially those that use instillation. Going back to this guy here, 500 years, I'm German, but I'm coming from Switzerland, therefore this is a Swiss guy. If I would be working in Germany, this would be a German guy. And if I would work in Austria, this would be an Austrian guy because he was working everywhere. But at that time, when he was a doctor in Switzerland, he mentioned exactly this, that the dose is making the effect. And now I make a next provocative statement. The Lee study. We have discussed today this Lee study, this older study, which I have not included here in the literature study from me. The Lee study, what would you say if I state the Lee study is a good study? You can use it. And the next provocative statement is the Lee study is a no-effect study. Because he has clearly demonstrated that 50 milligrams per cubic meter and less have no adverse effect and do not induce cancer and tumors. They are not higher than the controls. I have seen only the data that have been shown today. So it is a no-effect study. Only the overkill, if you go dramatically higher, then of course you get an effect. So for me, the Lee study is a no-effect study. You can discuss this later. Okay, those metrics may be revisited. I demonstrate this on an in vitro example, but you have the same in vivo. So you probably know the work from Klaus Wittmark. It's in the former institute where Helmut was in Munich. And he mentioned this this particle effect in vitro. If you apply particles in a suspension, if the density of these particles is higher than 1, then of course the particles start to sediment and you get a coverage of the whole cell layer. You see here in the middle a typical effect. 9 millimeter high is the medium in the cell culture. The concentration is 27 microgram per ml. This is in vitro is a normal concentration that many studies use. And you see after 8 hours, and after 24 hours it's much worse, you see that the bottom of your culture disk is covered by titanium dioxide. Okay? Keep this in mind, 27 microgram. And his conclusions is that most of the studies, that in vitro studies, that come up with toxicity of titanium dioxide or other particles are related to that you cover and bury your cells under a layer 65 nanometers high below nanomaterials if the density is higher than 1. Okay, so it's not a toxic effect of the material, it's a toxic effect because there is not enough oxygen, there's not enough nutrients, there's not enough molecules that the cells need, there's not enough interaction with the outside anymore because they are covered and buried below this. I give you an example here. This is the experimental setup. You have your dispersion, and if you wait long enough, you have buried your cells below. And there's more than this. There's more confusion about concentration and dose. The first is, if you look carefully into the publications on, especially in vitro, you will find that they give, for instance, microgram per milliliter. But look carefully if they give the number of milliliters in the culture disk at the end. Mostly you are not aware of this and you don't find this. So you don't know how much they really applied. The same is true for microgram per square centimeter. If they give the numbers in microgram per square centimeter, then it's dependent on the confluency and the number of cells that you have in your Petri dish. So it differs tremendously between the different experiments. And as I mentioned before, if the density of the material is larger than 1, most of the particles sediment, and you have a much higher concentration above the cells than in the overall volume. And the other side is true as well. If you have a density less than 1 or around 1, normally these particles are floating around and then the cells will never see. So these are mostly declared as non-toxic. Okay, this is based on these data. You see 27 microgram per ml, and you have 25 microgram per centimeter square, and this is more than 60 nanometers a layer above the cells. So here is only one example. I can give you hundreds of examples of this. Only one example, you see here concentrations used in a study, 50, 100, and 200 microgram per ml. It's the minimum concentration is double the concentration that Klaus Wittmark mentioned with these 65 nanometers layer above. So, of course, these cells have problems to survive, and of course, you find at the end a positive comet assay after 48 hours. Okay? So, this is in vitro. Now I have stolen some slides from Günther. Thank you very much, Günther. He has shown this study here from Wang et al. It's in vitro, it's intranasal instillation of titanium dioxide. And if you look very carefully into the study, you find that they have made bolus instillation into mouse of 7.5 milligram, which reflects 17.5 grams into human nose. It is a real snuff. Try to get this into your nose. You are laughing, but it's a very severe point. Most of these studies are done in the same direction. Most of these studies use exactly these concentrations. It makes no sense. Okay? Therefore, David has asked, I invited him to give a paper in my journal where I'm a co-publisher, a co-editor, and he asked this question, how meaningful, on the one hand, are risk determinations in the absence of a complete data set? And the next is, of course, he raised the question of no-effect studies. So we have, in all these 25,000 studies that are published, mostly the people demonstrate a biological effect. Because otherwise, only in rare cases, they will not get it published. So we have a totally distorted view. The regulators have a totally distorted view about the toxicity of nanoparticles because published is only positive effects. And they put, mostly these are masters or PhD students or postdocs. Of course, they have to deliver some effects, so they put more and more until they reach exactly this scenario that they can measure something, and then they can publish it. Therefore, it's very important to publish no-effect studies. because this cannot be the future. We know that labeling in the EU for food and for cosmetics is already in place. And you see here, there is not titanium dioxide nano labeled. It is no titanium dioxide is labeled. And this is, of course, nonsense. But this is a company. So you see that companies make nonsense as well. Okay. Some extra slides. I have 4 minutes left, I see. Some extra slides because I put them in. We had this discussion before. We had this discussion, and it's probably for tomorrow for the genotox. It's important as well because we discussed this interaction of nanoparticles directly with the DNA and the movement into the nucleus. Because nanos are so small, they can go into the nucleus. No, it's nonsense. They don't. We have a nuclear pore of 39 nanometers, but the nuclear pore is closed. We have a protein sitting in the middle of the nuclear pore and controlling exactly what is going through and what not. And it opens only for the material that the cell wants to go out and in. And you can demonstrate, and there are some very nice papers to demonstrate this, you can demonstrate a nanoparticle going into, if it's smaller than 39 nanometers, into the nucleus if you bind a nucleus signaling sequence on it. Then you can direct a nanoparticle into the nucleus. It's demonstrated. But if you don't have this peptide on the surface of this particle, it will never go into the nucleus. I have looked for it. You see here the selection. First, every cell will take up nanoparticles. I have mentioned it already. You only find a few numbers of exceptions. And you see here, for the nucleus, for those that have really looked for, if they are distributed equally in the cell and to the nucleus, you find the majority says no. You have heard Paul Baum. And there are some, more than 30, that say yes. I have looked very carefully into these papers. I will give you only one example. And you see, we have not discussed this earlier, Paul Baum and me, but we choose the same paper for demonstrating how titanium dioxide is going into the cell and where it is in the cell. And I promise you, we have in our lab made thousands of slices, thousands. And never we have found a single particle inside of the nucleus. So it's always looking like this here in lysosomes. So taking the same cells, the A459 cells, these are lung adenocarcinoma cells, alveolar cells. You see another study. They have used these particles here, and they delivered in their publication this picture. This is the picture of the material they have used. The titanium dioxide, there are particles, there are some rods, cylindrical things, and you see the scale. Yeah, so these are not so small particles. And you see on the right, they show the particle distribution equally over the whole cell, in the cytoplasm as well as in the nucleus. And they point it out with the black arrows. But you see that these particles are much smaller. No, these are HepG2 cells. These are not A45 cells, but it doesn't matter. But this, I draw them in here in the same size as they have delivered it, and you see that it could not be. These dots, these little black dots, they show and they discuss as titanium dioxide to be in the nucleus. This cannot be. It's not possible. It's not the same material. Otherwise, they have used other materials, a smaller particle or something like this, so it's not possible. What is possible is that such a very, very continuous location of particles in the cell is normally an artifact by contrast materials like lead or uranyl acetate that they use for the transmission electron microscopy. So these are contrast, normally these are contrast media that you use, and these, if you don't use the appropriate method, they sediment, these contrast particles sediment, and then you have exactly this equally distributed particles on the surface. And if you look carefully in all these studies that show a nuclear location of these nanoparticles, you find that they all have these shortcomings. This is a very good study done in Bern, at the University of Bern, with a tomograph, with an electron microscopic tomograph. They can make 3D slices and 3D calculations, and they counted each nanoparticle in the cells. And you see here, these are gold particles, plain gold and pegylated gold particles, and you see these are much smaller than the nuclear pore, and you see that these particles all are in lysosomal vesicles and never have been found in the cytoplasm, never have been found in the nucleus, and never have been found in mitochondria. Okay, this is the extra slides. Now the conclusions at the end of my talk. So far, in all these 6,000 papers I found at the end, and of course for titanium dioxide it was much less, there's no evidence for a nanospecific effect. In vitro, the overwhelming majority of the studies does not compare their results and does not carry out experiments with a respective bulk material or with the ionic form. In the case of zinc oxide and copper oxide, it makes sense to use the ionic form as well. So they cannot say this is a nanospecific effect because they don't have done the control experiment. And in vivo, it's the same. And all in reliable data in vivo, they point to that for titanium dioxide, it is a normal dust particle effect similar to the GBP. For the genotoxicity, it's the same. All studies showing positive results are pointing to secondary genotoxicity via inflammatory or oxidative stress phenomena. And most of the studies, I should say, do not include adequate controls. This is a very, very big issue. I don't know what the reviewers are doing. Most of the studies should have been rejected. Because if you cannot judge if this toxic effect is a severe effect or not, it makes no sense. So therefore, you should have a minimum, the negative control, a positive control for your endpoint, and then your materials. The materials probably with a reference material, with a known toxic material, with a known non-toxic material. So all these appropriate controls are missing. And therefore, you cannot do any classification for severe, minor, harmless, or hazardous. And next, to my knowledge, there are no in vitro studies that show a severe or strong effect of titanium dioxide. All these are, if you go from 50 microgram up, all these are overdosed. And there's no real demonstration of a DNA damage. And the same is true for the in vivo studies. If there is a genotoxic effect, it's secondary. It's dependent on oxidative stress and inflammatory conditions. And thus the OEL that we already have for the nano titanium dioxide of the suggestion of 0.3 may be good for the workplace, may be justified. It's not me to justify it, but maybe. But for me, in all these studies, there's no objective reason for further reduction. No. Okay, with this and the view of the mountains of Switzerland, I thank you for your attention.
Okay, your opportunity to ask questions, come up with comments. Yes.
No, just a remark to the high dose effect. You know, in most of the toxicology journals, the reviewers, and we all agree, would accept that if there is a dose response and indicating where the no effect level is, and by that having certain different doses, and with some kind of indication of the human relevance of the findings. Mm-hmm. You know, that's what we insist in order to accept such information.
Thank you for this wonderful comment, Helmut, because in most of the studies I have looked at, in these 6,000, you find a real good dose response curve in very rare cases. So very often you cannot look for the no effect level.
Okay.
There are many single concentration experiments, in vitro as well as in vivo. There are many single concentration studies published. What is the sense? Please say. You are experts as well. What is the sense of a single concentration study?
Yes, so not all of you can see the hand gesture that was made as a result, but let me translate your hand gesture. Eliminate, right?
Sure, yeah. These are not relevant.
Yeah.
These studies, forget them, you know.
Yeah. But the title says toxicological study on.
Okay, 2 more.
Hi. Can I just make a comment on the genotoxicity? Because you mentioned the COMET assay, and people who are not that experienced in genotoxicity tend to think that all of the different endpoints are equally relevant in terms of predicting cancer, but they're not. And there is an OECD overview document which was published recently which clearly states that indicator tests, so those that are very upstream, very early events, and the COMET assay is a DNA strand breakage test, those strand breaks may be lethal, may be effectively repaired, may not turn into heritable changes in the genome. So we should not give those kind of results the same weight as we would to a test that measures stable, heritable changes such as gene mutations or chromosomal damage. A lot of the tests that have been done on titanium dioxide are DNA strand breakage tests.
A wonderful comment as well. And I showed you this experiment set up, these columns with 50, 100, and 200 microgram per ml. And they did the COMET assay. And we did it, of course, as well in our lab with totally different materials as well as with titanium dioxide. And if you use 50 microgram per ml, like they did for the COMET assay, first, you cannot do it objective because you see the particles. You cannot avoid the particles in your gel. You will extract them with your cells and with the nuclei to the gel. So you see which of these slices are treated and which are not treated, first. And second, at 50 microgram per ml, you have so many particles in it, they interact in the gel with the DNA, and they collect it. And you have brilliant spots in your nucleus. And if one of these spots is walking in the gel, you find a tail. So it is not doable with such concentrations of nanoparticles.
Okay.
Just a brief comment on your complaint that you cannot understand that reviewers can accept these high concentration studies. And the reason is because most of the reviewers do it themselves that way.
Yeah. Okay.